The protein folding problem has been called one of the great unsolved challenges in biology. A protein is a chain of amino acids, anywhere from a few dozen to several thousand of them. And when that chain is released into the cell, it folds into a precise three-dimensional shape in a fraction of a second. That shape determines everything the protein does. Get the shape wrong, and the protein fails, sometimes catastrophically. The puzzle is, how does it know which shape to take? The chain has an astronomical number of possible configurations. If it were sampling them randomly, even at the fastest possible rate, it would take longer than the age of the universe to find the right one. Yet it folds reliably, rapidly, and correctly every time. The standard model treats this as a search problem and devotes enormous computational resources to simulating it. Wave physics dispenses with the search entirely. The chain does not search, it assembles. Each amino acid is a relatively rigid geometric object. Its binding sites, the places where it connects to neighboring amino acids and where it can form bonds with other parts of the protein, are oriented at fixed angles determined by the nuclear knot architecture, exactly as we discussed in the chemical bonding video. Those angles are structural facts about the atom, the same way the angles of a machined bracket are facts about the bracket. When two amino acids are joined, the membrane enforces an additional constraint. The bond between them must be planar. There is only one orientation that satisfies this constraint, and because each subsequent amino acid faces the same fixed geometry, the chain does not explore a space of possibilities. It follows a path determined entirely by the sequence of amino acids and their preset orientations. The protein folding problem was not hard because the search space was large. It was hard because the field was looking for a search. There is no search. The geometry determines the fold. The clearest demonstration of this is sickle cell anemia. Normal hemoglobin carries a specific amino acid, glutamic acid, at position 6 of the beta chain. Sickle cell hemoglobin carries valine at the same position. That is a single substitution out of hundreds of amino acids. The difference between glutamic acid and valine is purely geometric. One presents a flat planar surface at its binding site, the other presents a rigid forked shape. In the deoxygenated state, that forked valine at position 6 slots precisely into a pocket on an adjacent hemoglobin molecule. Not approximately, precisely. The contact surface buries 182 square angstroms with essentially zero strain, and only one docking orientation is geometrically available. This click lock nucleates a fiber. The fiber distorts the red blood cell into a sickle shape, and the result is the disease. The entire pathology of sickle cell anemia is a single geometric event with an inevitable consequence. Replace one planar surface with one rigid fork, and the protein locks where it should not. It is a result of physical cause and effect, not probability. In the next video, we will discuss minting new matter,